So after these two first very high-level talks, we are going to change gear and to move to a very low-level, a deep dive into the data. Mass-spectrometry-based single-cell proteomics is a young and really exciting field that comes with many challenges. Great progress has been made, of course, and this conference is a demonstration of the successes and promises of the field. And so in this presentation, we would like to share our efforts in proposing an infrastructure for standardized and reproducible analysis of these single-cell data. And to illustrate this infrastructure, we will present a replication of the analysis of the recent Scope 2 MS data by Harrison, Specht, and colleagues. So I'd like to start by emphasizing the importance and the value of replication. Now, of course, here I'll be focusing on computational replication. Everything we do with start from low-level quantitation data, so PSM data. And I'll posit that results that can be reproduced are more trustable. So what I show here is the expectation. We have some data, we process it, we analyze it, we get results, and eventually these results lead to discoveries. But the reality is that many steps along the pipeline can go wrong, and very often will go wrong, leading to inaccurate, misleading, and wrong results. And so a pipeline that can't be reproduced is a pipeline that is ill-defined, that leads mostly to false discoveries. Now, of course, it's really important to emphasize that replication isn't a guarantee for accuracy, of course, but an analysis that can't be replicated is one that can't be trusted. So replication is the first step towards trustworthy and shareable analysis. Now, there is a second value in replication that's a more kind of selfish one. The development of non-trivial software tools isn't done out of the blue. It's not done in isolation. Ideally, it's collaboration between the developer, the bioinformatician, the person that produces the data, and eventually, of course, the user. And so I think that a replication or reproduction-based development is actually a really efficient way for tool development. And so, as I mentioned, replication is the first step towards defining some data infrastructure and is the base for further improvements. Now, why do we start to focus with Scope 2? Because the goal, of course, of our tool is not to focus on a single technology. And indeed, many of the processing steps that will be mentioned later are ubiquitous to any quantitative data processing. But Scope 2 offers an ideal reproduction-based development use case. The full protocol is available, the data and all the scripts were available, plus we got outstanding support from the authors. And so here are our objectives. We want to contribute in standardized and principled data analysis that is broadly applicable for mass spectrometry-based single-cell data. We want this to be open, transparent, and reproducible. A computational infrastructure that allows this is extremely important because it can be leveraged to further improve data analysis, modeling, and interpretation. And knowing my background, of course, I believe that iBioconductor is an ideal platform to do this. And all this has been implemented in a new package called SCP. So I'm going to start to present the infrastructure behind SCP, because SCP relies on other existing packages. And the first one is QFeatures. That is a general infrastructure, so it's not specific to single-cell proteomics, that allows to manipulate quantitative data from mass spectrometry experiments. So proteomics is multi-level, data is acquired at the spectrum level, that's the low level, and then protein level data is progressively built up through multiple processing steps. And the goal of QFeatures is to explicitly record these successive steps so that the user can navigate up and down the different levels. So here's an example based on real data. So we have two proteins, STAT1 and STAT2 along the lines, and these three levels, proteins on the right, peptides in the middle, and PSMs on the left. And so it's very easy at the end of an analysis to want to check some proteins. And it has to be easy to go back to the peptides and the PSMs. So for example, if we look at step 1 and step 2 that have very similar profiles along these 10 samples that were assayed, we can see that step 1 is based on a single peptide and a single PSM, and step 3 is based on single peptides, and here we have 9 peptides, yes, and 10 PSMs. And we can assess visually easily, and this of course can also be computed, the variability at these different levels. Now there is a second really important part of the SCP data infrastructure, and that's the SingleCellExperiment package from Bioconductor that has been developed by some of our colleagues working on single-cell RNA sequencing. And the figure here represents the data class that supports the single-cell data analysis. So we have one or multiple tables for the quantitative primary or transformed data. So here we have features, so this could be genes, proteins, peptides along the rows, and different cells along the columns. We also have a table that captures metadata for the rows, and we have another table that captures metadata along the columns for the different cells. And another important slot here is shown at the on the right-hand side that allows to store the results from various dimensionality reduction techniques, which is a really important widely used tool in single-cell analysis. And so the infrastructure for SCP is the following one. We have a set of single-cell experiments at the PSM level, for example, different batches, at the peptide level, at the protein level, and these different quantitative assays are all collectively linked and managed by QFeatures. And then, of course, and that's quite important, in addition to that, SCP provides the functions that allow us to process and analyze single-cell proteomics data.
Okay, so to start the replication, we have first formatted the supplementary data provided by Scope2 preprint using the SCP infrastructure that Laurent just introduced. The data set is actually stored in a data package called SCP data. So we first load SCP data with line 1, and we then fetch the data using line 2. The show methods gives us a compact overview of the data set. And so you can see here from the header, the data set is a QFeatures object containing 179 assays. As Laurent mentioned, every assay in the data set is a single-cell experiment object. The assays 1 to 177 contain the quantitative PSM data and the metadata for the different Scope2 sets. They contain either 11 or 16 columns, depending on if TMT11 or TMT16 protocol was used. The before last assay is contains the peptide data for all sets, and the last assay contains the protein data. So this the tabular data that is generated by MaxQuant or Proteome Discoverer can easily be converted to such a data frame using the read SCP function. The sample metadata that is common to all assays are stored in a single table. We call it the core data. Each row represents a sample with different associated fields, metadata fields, of course. So in this example, we have a field that is called set, which gives the the Scope2 set name. We have channel, in which the sample was acquired. We have the sample type to give functional annotation. We have chromatographic batch, and etc. All the useful information that is required for an analysis is all contained in a single object ready for processing. So the Scope2 analysis is composed of a series of data processing steps, and I already mentioned the loading the data. So we start with the PSM quantitative data for 177 batches. Next comes PSM filtering, dividing the expression channel by the reference channel, PSM to peptide aggregation, joining the different sets, single cell filtering based on the coefficient of variation, normalization, removing of highly missing peptides, and log transformation. After this, we get the peptides dataset that is provided in a preprint. This peptide dataset will further be aggregated to proteins, normalized, imputed, and batch corrected. And then we get the final datasets, the protein dataset that is available online. So I will not have time to cover all the steps, but I will show just a little, well, those representative ones. So let's see how all of this works. One common functionality is to remove low confidence features based on the metadata that is stored in the dataset. So for example, we could remove the PSMs that were matched to the reverse database that serves as a decoy database. This information is actually already available because it was computed by MaxQuant and is stored in the reverse field of the metadata. So using the filter features, we can perform this. We supplied the data set, and we have to give it a condition. In this case, the features for which the reverse field or the reverse data is positive are removed. So the function then after returns a data set containing only the PSMs that passed that condition. Of course, not all metrics are, well, yes, some QC metrics might not be computed by MaxQuant and are let's say, specific to single-cell proteomics. So for instance, scope 2 computes a sample to carrier ratio, which allows us to discard samples with intensities that are higher than expected. It also computes a peptide FDR, which is the expected proportion of features wrongly assigned to a given peptide, or also the cell median coefficient of variation. It's basically filtering out cells for which the reliability of the protein quantification in a cell is too high. Sorry. So you can see here we can perform this using the compute median CV. It takes the data set. I is the assay to which we need to compute this median CV, and then it takes additional columns required for the computation. The median CVs are afterwards, so after running the function, are stored in the data set. So this is very handy actually, because you could use this stored information for further plotting or subsetting the data. So here I give you a little example for plotting. On line 2, you can see we first extract the assay we want to plot, so where the data is present. We extract this metadata using the call data function and format it to a data frame, which allows us later on plotting using the ggplot functionality. We can see from the graph that the blank samples, so in red, show much more variability than the single cells, so blue and green. And the black line is used in the scope 2 to filter out those cells with high coefficient variation. The next step, another step, is the feature aggregation, which includes combining feature data into a higher level structure. So for example, we can think of aggregating peptides to protein. This requires 2 steps. First, we combine the quantitative data from multiple peptides to a single protein. And second, we store the relationships between the aggregate peptides and the protein. We use for this aggregate features. It takes the data set, it takes the assay that needs to be aggregated, the name of the newly aggregated assay, so in this case proteins, and it also requires the metadata field, which tells to what protein a peptide belongs to. Finally, we need to supply it a function that will allow to combine the data, and eventually with possible additional arguments. Another important thing is managing missingness. So single-cell technologies contain many zeros, and so does, of course, single-cell proteomics. Zeros can be either biological zeros or technical zeros, and they are better replaced by missing values, or NA. This will avoid confusion in downstream analysis. This can easily be done using the zero is NA function, where you just have to give it a data set and the name of the assay you want to replace zeros. We can also remove highly missing features. So for instance, we can remove features if it has more than 99% missing data. This is performed using the filter NA function, and you can, well, the PNA argument will control the tolerated proportion of missingness. Finally, missing data can also be imputed using the impute function. So you can supply again the data set, telling which assay do you want to impute. So in this case, we want to impute the assay with proteins using the k nearest neighbors method, using k equals to 3. Other methods than k and n are also available, and this facilitates benchmarking of different imputation methods. So next we have data transformation. So a common data transformation can be easily applied, such as log transformation or normalization. And here I give you an example for log2 transformation. So we need again to supply the data set. We supply again the name of the peptide we want to transform. We give it also the log base, so in this case log2, base 2. And we give the new name of the processed data, the processed assay. And finally, we can also apply custom functions. Our framework can easily be adapted for that. In order to do this, let's take an example. Let's say we want to do batch correction using the combat algorithm. There is a 3-step procedure. So first, we extract the assay from the data set and store it under X variable. Then we can apply combat to the data, but it requires a data matrix, and this can be extracted using the assay function. I also show a few dots because there are additional arguments, but I will omit them for clarity. So once we have overwritten this data matrix, we can add it to the data set as a new assay, and of course apply it a new name. Remember that the assays we add to our framework should be single-cell experiment objects. So we replicated the scope 2 analysis using our standardized framework. The results will be shown in the next slides, and we always compare the scope 2 expected results to our output. So in this case, we first compared the selected features from both datasets, and we can see that for both peptides and protein assay, the overlap is very high. Around 9,000 peptides are common for both workflows, with over 99% agreement, so almost perfect. Similarly, for proteins, around 2,700 proteins are common for both pipelines, with a 97% agreement. Next, we also can look at the numerical comparison. So the graph is, the two graphs show the distribution of the differences between the two data sets. In order to do this, we had to subset for the common features and the common cells. The distribution for both data sets are sharply peaked around 0. Only for the protein data, it's a bit more spread out because the differences that we see already at the peptides tend to accumulate during the processing. Even if there are small differences, well, I mean, although there are small differences, the data sets are still highly similar. This can be seen from this weighted PCA plot. The left figure is actually the figure 3A from the preprint, and the right figure is our attempt to replicate it. The two PCA exhibit exactly the same trend. So this indicates that the underlying main patterns are well replicated. I'll finish my part with a small note about reproducing data analysis. Identifying key processing steps is essential for reproducible work. So good software development includes continuous maintenance and improvement. And small changes in the implementation of an algorithm can impact the reproducibility of an analysis. For example, we noticed, and with the help of Harrison, that the batch correction results using Combat may differ depending on the software version. These differences are due to a new environment feature filter, and this was added recently to the batch correction algorithm. And this filtering step, in turn, highlighted technical artifacts of the data. Okay.
Okay, thank you very much. So let me conclude this talk by highlighting our main take-home messages. So I think we are all here because we are excited by mass spectrometry-based single-cell proteomics. But we are, of course, all well aware of the challenges. And with SCP, we hope to address the computational part of these challenges by providing a principled and reproducible data analysis framework to manage the data, to analyze the data. Now, as I already said, SCP is not specific to scope 2 or labeled TMT data. It's also applicable to other protocols like the nanopods that use labor-free quantitation. And as Christoph has highlighted, many of the processing functions that are already available are applicable to many pipelines. I think another point that is important to highlight is that SCP uses this SingleCellExperiment package from Bioconductor that is also used for single-cell RNA sequencing analysis. And I think that's very useful because it will facilitate applying, when relevant, the same processing methodologies to single-cell proteomics and single-cell RNA data, and so to kind of facilitate the integration of these different modalities. And I think the last point is absolutely crucial. As I said in my introduction, being able to reproduce an analysis pipeline is really important, at least according to us. But really, it's only the first step. This is only the beginning of our work, and we are already relying on SCP to develop the next generation of data analysis functionality for these type of data. Most of these resources are already publicly available. So if you go to that first URL there, you'll find a website for SCP. So there is still a lot of work to be done, including adding more documentation. That's what we are doing at the moment. But you're already free to try it out and ask questions if you have issues. But we'll be adding lots of documentation in the new feature. So the scope MS2 data that was used for this replication will also be made available in another data package called SCP data, and we will also add other single-cell proteomics data sets that we will also use for our development. That package is not quite ready yet for technical reasons, but hopefully it'll be ready within a couple of weeks. The QFeatures package has been around already for quite some time, and it's part of the R4 mass spectrometry effort. And so if you go to r4masspectrometry.org, you will be able to find a link to the QFeatures that already comes with plenty of documentation, and QFeatures will be available from Bioconductor very, very soon. And finally, the SingleCellExperiment, also available from Bioconductor. And if you go to that URL there, you'll have access to our slides. So I'd like to finish by acknowledging our funders that have made this work possible, but also people from Nikolai's lab, in particular Nikolai, Harrison, and Ed, that have been really, really helpful in helping us to go through the analysis pipeline, explaining when we didn't quite understand a few things, but also were also very active, proactive in kind of listening to our comments we had about their analysis. So thank you very much for your attention, and we would be very happy to answer any of your questions.